Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So welcome to my session. So today we are talking on a new topic, that is neural control and coordination. So we'll be discussing about the structure of neuron, being the first lecture today. So we are almost towards the end of human physiology. So we are done with digestion, breathing, body fluids, excretion, locomotion. So right now we are here talking about neural control and coordination, right? Hi Natasha, yes, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay, so let's talk about neural control and coordination. So this is a very interesting chapter and it's a very important chapter as well. So here we'll be talking about structure of neuron. So in the next lecture we'll be talking about the structure of human brain, human eye, and then we'll end it off in style in the human ear. Alright, ready for today's lecture? So, well, I could not have or come for yesterday's class because I was occupied with some other work. So, we'll continue with sexual reproduction. Don't worry, I will teach you the entire chapter of sexual reproduction. No issues, okay? Yes? Done. So, I hope your preparations are going on well. Yes, everyone is concentrating because today is the last day of July. So done and dusted. You only have 30 days, almost 31 days plus another 10 days. So another 40 days you can take into account. So 40 days is too less of a time if you say, because you have to be peaceful as well. You have to be relaxed as well. And you have 3 subjects, so physics, chemistry and biology. I hope you're doing well and your preparations are going on fine. Okay? Yes? Chalo, theek hai. So now let's begin with neural control and coordination. So let's talk about the structure of neuron. So please don't forget to like, share and subscribe and share the link with your friends so that your friends will also join our session. Yes, Shimpi, hello, a very happy good evening. I'm good, thank you. How are you? How is your preparation going on, Shimpi? Your school classes are also going on? Yes? So Shimpi, please don't forget to like, share and subscribe. Okay, so we are talking about the most important chapter, the most interesting chapter in human physiology, that is your neural control and coordination. So let's talk about the structure of neuron. Okay, yes, so here we go, structure of neuron. So before we go into the structure of neuron, so I have few announcements to give you. Hi Munna, so you're also well very early today, huh? You also came along with Shimpi. So thank you for joining my session. So your name is Pallavi, is that so? So thank you for joining my lecture. So please do like, share and subscribe. Yes, I am super good, Munna. Okay, I'll call you Munna. Yes, how are you doing? Is it raining in your place? Because yes, it is raining in Kota. The weather is very pleasant here. Okay, how is your place? I hope things are going good. So you and Shippy do not fight any further. Okay. So before we go into the structure of neuron today, I have an announcement. So we will meet directly on Wednesday because Monday, Tuesday I have some work. So Wednesday we will have a mega mentee. So it will have 40 questions and the winner will get access to a free mock test series. So please prepare well for that mentee. So the chapter or the unit is the 3rd unit of class 12, that is genetics and evolution. So you have principles of inheritance, molecular basis of inheritance, then you have evolution. Right? So you have 3 chapters and I'm giving you 3 days time. So please prepare well for the mentee. So it is mega mentee. So now please don't take your mentee lightly because in a small population you know where you stand. So you have to be very alert, very determined, very focused. So please prepare well for the mentee. So I will meet you directly on Wednesday after this. So Wednesday 5 PM we are having our mentee quiz. Okay? Hi Anthony, yes, a very happy good evening. So welcome to my session. So please do like, share and subscribe. No, I'm not there in any social media. Okay, so the unit is genetics and evolution and the time is 5 PM. Alright, so this is the announcement of mega mentee. It is on Wednesday, that is 4th of August. We have a mega mentee. It is from this unit, genetics and evolution. So here we have principles of evolution, molecular basis, principles of inheritance, molecular basis of inheritance, and then we have evolution. So all the 3 chapters, so you're going to have a mega Menti. It's going to be for 40 questions. So it's going to be a bit tough because I have taught you a lot in detail in principles of inheritance. So please go through everything. I'm giving you 3 whole days time. So we will be meeting on Wednesday at 5 p.m. Don't forget, okay? Yes, so prepare well, join for my Menti. So the Menti might go on for 1 hour, but we will play slowly, but we will play strongly. Yes, done everyone? Okay? Okay, chalo. So now let's talk about your neuron. Fine. So lecture 1 of neural control and coordination. So what does this chapter has to tell us? Let's talk about in detail. So we are going to talk about the human neural system. So how is the human neural system, the architecture? So we are going to talk about that. So we are going to talk we'll talk about the neural system in general, starting from your animal kingdom, how it started. So whether they have a well-defined neural system or not. Okay. So then we'll talk about human neural system. So how is the human neural network designed? So then we'll talk about the most important topic, neuron, the structural and functional unit of the brain, right? So what are its structures? So I guess you remember we have 3 main structures. So this is your dendrons, axons and your cell body, right? Okay. Did you see Tokyo Olympics? I just see the news at the end. So who won, what medal we got? So it's not that I watch it fully. No. Yes, what information you want me to know? Yes, what is it, Antonia B? So guys, please do like, share and subscribe. So please do like the session. Okay. So then we'll be talking about the central nervous system. So this includes the structure of your brain. So then we'll talk about the reflex action and the reflex arc. So then we talk about the sensory organ, that is your ear and the eye. Fine? Okay. So let's move forward. So we are going to talk about the neural system. How well is the neural system defined? So neural system of all animals is composed of a highly specialized cell that is known as neurons. Hi Rani, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Yes, I saw a message. Yesterday's class had to be cancelled because I was busy elsewhere. So last minute the work didn't get over, so we had to pull off the class. So yes, I'm here today. Okay. So don't worry, I will continue sexual reproduction and flowering plants from Thursday. So Wednesday we are having a Menti quiz, Dharni. So please make a note of it, okay? So today PV Sindhu lost her match. Yes, I saw the flash news. Yes, that is really sad, right? Okay. Okay, so we are talking about the neural system. So the neural system of all animals, so whichever in lower organisms your neural system is, so it has a specialized type of cells that is known as neurons. Okay. So neurons are the ones which they are the superheroes when we talk about your central nervous system. So they detect, receive and transmit different kinds of stimuli. What do? So they deduct, they receive and they transmit different kinds of stimuli, right? So for example, let's talk about Hydra. So Hydra belongs to which phyla? Yes, so Hydra belongs to phylum Coelenterata, if you remember. And they have a polyp-like structure. So what is a polyp? So it is a cylindrical structure like this and they are sessile. So it also has a network of neuron, okay? So insects, when you talk about insects like your cockroach, so we are talking about phylum Orthopoda. So this also has a brain. It has number of ganglion and your neural tissues. So this is present in your lower insects, okay? Yes, what is your doubt in that, Dharni? Yes, we have OBC creamy layer. Yes, so you have doubts regarding that? Yes? Okay, so in insects we have a brain, okay, not a very well developed complicated brain, but we have a brain. It is made up of ganglion and neural tissues. Done? So vertebrates have well developed neural system. So we belong to phylum Chordata. So under that we belong to subphylum Vertebrata. Under that we belong to class Mammalium, and we have well defined neural system. So this is your Hydra. You can see the Hydra is made up of a structure or network of neurons, right? So this is how the neural system is developed. Yes, I will get back to you in Telegram. I will approach your doubts. Okay, Rani? Fine. So this is the base. So this is how it all started. You should know the story. So talking about the human neural system. So the human neural system is divided into two parts. One is central nervous system and the other one is peripheral nervous system. Okay? Yes? So now talking about your central nervous system. So central nervous system is brain and spinal cord. Okay. So PNS, peripheral nervous system. So this is all nerves of the body are associated with the CNS. So CNS mainly talks about your brain and spinal cord. The interconnections between them, that is the nerves, it comes under the PNS. Okay. Yes, Swami. Hello, hi. A very happy good evening. Yes, welcome to my lecture. So please do like, share and subscribe. Alright, fine. So CNS is brain and spinal cord and PNS is all the nerves of the body that are associated with the CNS. Fine? Okay. So this is how it is. So moving forward, so we will talk about CNS. So CNS is the whole nervous system, okay, the central nervous system you can say. So it is derived from the embryonic ectoderm layer. So heart is derived from mesoderm. So your connective tissues are derived from mesoderm. Your muscular tissues are derived from mesoderm. And your neural cells or the whole nervous system is derived from your embryonic ectoderm. Right? Yesterday you were taking class. No, yesterday I had to call off the class. I was busy elsewhere. So yesterday's class I will reschedule it somewhere in next week. So next week we'll have 2 lectures somewhere so that we can finish off sexual reproduction. Alright, Swami? Yes? Okay. So the whole nervous system is derived from the embryonic ectoderm. Alright, so the CNS includes your brain and spinal cord. So it is formed. So this brain and the spinal cord or the central nervous system is formed from the neural tube. It develops from the ectoderm after the gastrula stage of embryo. Yes, we also have some idea about the gastrula stage. So gastrula is the stage just after implantation. So where you have the 3 embryonic layers coming up, ectoderm, endoderm and mesoderm, right? Okay. So it develops from the neural tube in the intrauterine life. The anterior part develops into brain and the caudal part of the neural tube into the spinal cord. So it is something like this. The anterior part develops into the brain and the caudal part develops into the spinal cord. Okay. So this is how it forms. This is your neural tube. This is how it is present in the uterine development. Okay. So we are going to talk about the PNS. So the PNS has 2 types of fibers, afferent and efferent fibers. So afferent is what brings information to the brain. So it is connected with the sense organs and it is connected to the CNS. So efferent takes the information from the brain. So your PNS is also divided into somatic and your autonomic neural system. So autonomic nervous system is further divided into sympathetic and parasympathetic, which always works in antagonistic fashion. That is, they work always in opposite direction. So visceral nervous system, part of the PNS where the whole complex of the nerves, fibers, ganglia and pulses are present. Okay, so that is your visceral nervous system. Okay, so this is about your afferent and efferent. So this is your PNS. Okay, so let me give you a recap of whatever we spoke so long. So we were talking about your neural system. So what is the development of the neural system? So what is the timeline? So we started with Hydra, right? So Hydra belongs to Coelenterata, phylum Coelenterata. So it has a network of neural cells, okay? It has a network of neurons. So this is the first development. So slowly we spoke about insects. So insects belong to phylum Orthopoda and class Insecta, right? So they have a not so well developed brain, but they have ganglial cells and they have neural tissues. Fine? Okay. So then we started talking about well-developed vertebrates. So we have a perfect neural system. So how is a neural system defined? How is it divided? So we are talking about the human neural system. So human neural system is divided into CNS and PNS. So CNS includes your brain and your spinal cord. Okay. So where does it develop from? Hi Shamita. Yes, a very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Okay. Right. So where does this develop from? So this entire neural system develops from embryonic ectoderm. Right? It is developing as a neural tube in the intrauterine life, the anterior part of which develops into brain, and the caudal part, that is the tail-like part or the posterior part, develops into the spinal cord. So this is how it is classified. This is your CNS. Okay. So the next one is we have PNS. So this is peripheral nervous system. So this is basically with the nerves associated from your CNS. Okay. So here we have two types of fibers. So the first fiber is afferent and the other one is efferent. So this takes informations from the organs to the CNS. So this is sensory neurons or sensory fibers. Fine. The next one is efferent. So this is taking information from the CNS to the sense organs. So this is known as your motor neurons. Fine. So apart from that, PNS also has two other classification. So this includes your autonomic nervous system and your somatic nervous system. So PNS, so this is associated with autonomic nervous system. So we call it as ANS. The other one is somatic nervous system. So this is SNS. So this is connected with the somatic cells of the body. This is connected with the involuntary organs. So your autonomic nervous system is divided into two. So one is sympathetic nervous system. The other one is parasympathetic. Okay. So sympathetic and parasympathetic always work in opposite direction. If sympathetic increases the heartbeat, this decreases your heartbeat, parasympathetic. So this is the entire classification of your human neural system. Is this clear? Yes. Is this clear? Understood? So parasympathetic will decrease your heart rate, whereas sympathetic will increase your heart rate. It will increase the cardiac output. This will decrease the cardiac output. Okay, Antonia B? Fine. So we are done with one part of the chapter, that is classification of the neural system. I have given you the tabular column. The next part is we are going to talk about the structure of neuron. So neurons are structural and functional unit of the neural system. So it is like this. So this is the structure of the neuron. It has three main parts. What are the three main parts of the neuron? So it has your cell body. Cell body is also known as cyton. Number 2, we have dendrites. And number 3, we have your axon. Okay, so we have three parts of your nervous system. Is this clear? Okay, so now let's talk about each of this in detail. So first part of the neuron, so we are talking about the cell body, which is also known as cyton. So cell body will have a cytoplasm, it will have a nucleus, right? So these are the features of a cell. Apart from that, it has small granules which are known as Nisser's granules. So this is also present inside your cyton. So we have what is known as Nisser's granules. So that is also present in your cell body. Okay. So the next part of the cell is your dendrites. So dendrites, so how do I define them? They are short, branched fibers which originate from the cell body. Okay, so they transmit impulse towards the cell body. They transmit or conduct impulses. This is towards the cell body. So this is the second part, okay. So now we are talking about the third part, that is your axon. So axon is the third part of your neuron. So this is the long unbranched filament, okay. So this transmits impulse away from the cell body. Okay, so this is axon. So we have cell body, we have dendrites, and we have axon. Okay, so the place on the cell body from where the axon arises, so this is known as axon hillock. So the space on the cell body from where your axon arises is known as axon hillock. Fine? Clear? Okay. So now the next part, the axon is like this, right? So this is the structure of your axon. Okay, so here, yes, I have dendrites like this. So these are my dendrons. So here I have my nucleus, right? So this is nucleus. So this part is my dendrites. Then this is my cell body or cyton. And this entire thing is my axon. So the portion from the cell body from where your axon starts, so this is known as my axon hillock. Okay, so here there is a small twist. So at regular intervals, your axon is covered by a sheath that is known as myelin sheath. Okay, at regular intervals, your axon is covered by sheath known as myelin sheath. So who produces this sheath? This is your Schwann cells. Okay, so myelin sheath acts as an insulator. So this is to prevent the impulses from leaking. Fine. So this is the role of myelin sheath. So the space between the two myelin sheath is known as node of Ranvier. Okay, done. So axon ends in axon terminal. So this portion is known as axon terminal. It starts with axon hillock and ends in axon terminal. Okay. So, we have knob-like structures or sac-like structures in your axon terminal. So, this is known as my synaptic knob. So, synaptic knob or synaptic vesicles, they contain or they release my neurotransmitters. So, this is the structure of axon. Is this clear to everyone? Yes, any doubt about the structure of axon? So, cell body is the first one. It is also known as cyton. Next is dendrites and the third one is axon. Right? So, this entire thing is my axon. So, we know cell body has a cytoplasm. It also has a nucleus. So, apart from that, it has what is known as granules, which are Nissls granules, right? Okay. So, dendrites, they are short branched segments. They also have Nissls granules, and this is long and this is unbranched. Okay, clear? So, never you should forget the structure of neuron. Clear to everyone? Yes, every single word that is written on the board is clear. No doubts? Shall we move forward? Yes? Okay. So, neuron, so part of the cyton from where the axon arises is known as axon hillock. So, some axon gives rise to side branches. So, yes, it is unbranched, but it has certain branches which we call them as collateral fibers. Okay. Hi Natasha. So, here this is known as an axon membrane. So, this is known as an axon membrane. The axon membrane is known as axoplasm. So this has a membrane known as axon membrane. So, this membrane also has a plasma membrane which is known as axolemma. So, this also has a cytoplasm known as axoplasm. So, just like we read about sarcoplasm and sarcolemma, so this is with respect to the neuron. So, neuron has a plasma membrane, that is your axolemma, and it has a cytoplasm known as axoplasm. So, remember these two, okay. So, that is what is mentioned here, okay. So, it ends in synaptic knob. So, synaptic knob is very close to the next dendrite. So, you have neurons arranged in a line. So, the gap between two synaptic clefts. So, there is a small space between the synaptic knob and the next neuron. So, it is like this. So, this is one neuron. So, here I have a next neuron. So, between the dendrites of the next neuron and the synaptic knob of the previous neuron, you have a small space that is known as synaptic cleft. Okay. So, that synaptic cleft has a gap of only 200 angstrom. Okay. Yes, sir. A very, very happy good evening. So, welcome to my session. So, please do like, share and subscribe. Okay. So, this is the structure of neuron. So, you understood what is synaptic cleft? It is the space between the previous synaptic knob and the succeeding dendrites of a neuron. So, that space is very small and it is for 200 angstrom. Okay. Fine. So, this is your synaptic cleft. So, moving further, so here we have based on the number of axon and dendrites. So, axon is one important part, dendrite is the next important part. The neurons based on the number of axons and dendrites, they are divided into three types. One is multipolar, the other one is bipolar, the third one is unipolar. So, these are the three different types of neurons which is also important. Okay. So multipolar is when they have many axons and many dendrites. So basically it has only one axon but it has many dendrites. So this is found concentrated in your cerebral cortex. So the type of neuron and the location is absolutely important, okay. So multipolar neuron is when you have one axon, that is long unbranched filament with many dendrites. So that is known as your cerebral cortex, okay. The next one is bipolar. So you have one axon, one dendrite. Bi is 2, right. It is found in the retina of your eye. The next one is unipolar. So this is 1. Uni is 1. So it is a cell body with only one axon and no dendrites. So this is found in your embryonic stage. So this is bipolar. It has one axon and one dendrite. So it can either have 2 axons as well, like this. So the next one, or the cell body can be in the center. So this is unipolar. So it has only one axon. It has no dendrites. The next one is multipolar. So if you can see, there are no dendrites here, right? But here you have a lot of dendrites. So this is multipolar. So you should remember the examples as well. So what are the different types of neurons we have? So we have 3 types of neurons based on the number of axons and dendrites. Okay, so what is the first one? So the first one is multipolar neurons. So multipolar neurons is when you have one axon, 2 or more dendrites. Okay, so example or where it is present in your cerebral cortex. Fine? Yes, Tamil Saran, a very happy good evening. So I guess you're new to my session, so welcome. So thank you so much for joining my session. So please do like, share and subscribe. Yes, Shubham sir, welcome to my session. So please do like, share and subscribe. Okay, so this is your multipolar. The next one is bipolar neurons. So bi is 2. So you have one axon and you have one dendrite. So this is bipolar. So example, this is seen in the retina of your eye. So this is bipolar, okay. The next one at the last, it is unipolar. So unipolar has only one axon and it has no dendrites. So it has no dendrites. So example, so this is seen in the embryonic stages. So this is inside the intrauterine development. So this is in the embryonic stage. So these are the 3 classifications of your neuron. Okay. So we are moving further. So we are talking about nerve fibers. An axon of a neuron is covered with 1 or 2 sheaths. We know that is the myelin sheath. On the basis of presence or absence of these sheath, the nerve fibers are again divided into 2 types. One is modulated or myelinated nerve fibers. So every axon which has the sheath is known as myelinated nerve fiber or modulated nerve fiber. Okay. So the other one is non-medullary nerve fiber where your nerve sheath or your myelin sheath is absent. Okay. Fine. So modulated or myelinated nerve fiber, neural canal in spinal, yes. So it is like this. So you have vertebral column. So inside each vertebral column, you have a hollow space that is neural canal. Inside that, you have your spinal cord passing. Okay. Rahul ji, yes, hello. So welcome to my session. Ma'am, now I am in class 12. Okay. Ma'am, I get frustrated attending 4 hours NEET classes, 5 hours school classes and I have to revise 11th to for 2022 NEET. Please suggest something. Okay. So this happens with everyone because now you have school classes as well, you have NEET classes as well, everything is online. Yes, so this is really irritating, right? So here it's neural. What is neural? So, okay, you are talking about the neural canal. So, neural canal is the spinal cord. Okay. So, no, neural canal is inside the neural canal you have the spinal cord. Yes, what is your doubt, Antonia B? Okay, so Rahul, so your frustration is valid. So, you have nearly 5 school classes and 4 NEET classes. So, in a day you are having 9 classes. So, how to cope up? Okay. And you have to revise 11th, so you haven't done 11th also properly. Okay, relax. So nothing is going to happen if you're going to get frustrated. So day is not going to become night if you get frustrated. Hold on, relax, breathe. Okay, so you are in class 12. So this is the month of July. So how many more months to go? So this is the 7 months. So 5 more months for December to come, right? So by December, make sure you're thorough with 11th and you're done with half of 12th. Okay, so plan in such a way such that if your NEET classes can be seen in recorded videos, keep them on hold. Keep them on hold. So go one by one. So watch a lecture, study the chapter in NCERT, solve questions, finish the chapter. So after 5 hours of school classes, make sure you sit down and study one chapter in physics, chemistry, biology from your NCERT. That is very, very important. So slow down for some time. So alternate days you attend NEET classes, alternate days you do self-study, because a student needs self-study equally. Okay? Yes? So this is very, very important. Alright? So relax, Rahul. This will really work. Hold on. So alternate days you study 11th. So alternate days, if you have the option of watching recorded classes of your NEET coaching, so make sure you will study alternate days 11 chapters and alternate days 12 chapters. So whenever you are watching your whenever you are studying 12 chapters, try to go in accordance with the class of your NEET coaching so that it is easy. Okay, Rahul? Yes? So please don't get frustrated. That is not going to help you. So here I said one word, neural. So I said unipolar is embryonic stage. So this is intrauterine. Okay, so this is the neural tube. Okay, so the neural tube here initially in the embryonic stages will have only one axon. Okay, Antonia B? Yes? Fine. So based on the presence, so we are talking about medullated or myelinated nerve fiber. So around the nerve, you have a sheath of fatty substance which is formed. So this is medullary sheath or myelinated sheath. Okay. So when you have a sheath of fatty substance, that is medullary sheath or a myelin sheath. Fine. Yes. Thank you so much, Rahul. I am happy that you found my suggestion useful. Okay. Fine. So the medullary sheath is not continuous, it is broken at certain points. So wherever points in which it is discontinuous, that is known as your nodes of Van Weir. Fine? Okay, so this is an insulating coating and prevents the energy loss during the conduction of nerve impulse. So myelin sheath plus the node of Van Weir is surrounded by a transparent cellular membrane, so which has your Schwann cells. Done? So it is like this. So we spoke about nerve fibers. So we spoke about neurons, right? So neurons were of 3 types, that is your multipolar, unipolar and bipolar. So this was based on the number of dendrites and axons. Okay. The next one is your nerve fibers. So this is basically your axon. So axon, based on whether it is surrounded by a sheath or not, the neurons it is divided into 2. So one is known as medullated or myelinated nerve fiber. The other one is non-medullary nerve fiber. Fine. So medullated or myelinated, so this has a fatty sheath surrounding the axon. Okay, so this acts as a layer of insulation to prevent the loss of energy during conduction of impulse. The next one, this is not continuous, this is discontinuous. Wherever the sheath is not there, that is your node of Ranvier. Okay, so this is myelinated. So this is when it has a myelin sheath. So myelin sheath is produced by a group of cells known as Schwann cells, which is present in the sarcolemma of a transparent cell surrounding your neuron. So that is where it is present. Okay, so now we are going to talk about the generation and conduction of nerve impulse. So it is already 43. Okay, so first let's talk about conduction of nerve impulse, and then we will move forward with transmission of nerve impulse. So neurons are excitable cells. Okay, so what are the neurons which are having a myelin sheath, which do not have myelin sheath? Okay, so the ones which have myelin sheath are the ones which are present inside your, exactly inside your CNS. Okay, so which does not have myelin sheath will be your homework. You will find out and tell me. Okay, so you will find out where non-myelinated nerve fibers are present. Is it present? Yes, it is present. You will find the example and tell me on Wednesday. Okay, yes, so that will be your homework. So neurons are excitable cells. So the membranes are in a polarized state. So how does the membranes have charge? That is because of the ion channels that is present. So these ion channels are selectively permeable to certain ions. So neurons in a resting state, so the axoplasm has more amount of sodium ions outside and more amount of potassium ions inside. Okay, so now negative charge is present in the axoplasm. So membrane is impermeable to the negative charges. So inside the membrane you have negative charges and outside you have positive charges. Okay, so thus a concentration gradient is maintained. So this is made possible by your sodium potassium ions. So it is like this. So I will explain, don't worry. I don't have a slide. Yes, I have a slide. So we are talking about the conduction of nerve impulse. So what is this? What is this conduction of nerve impulse? Suppose I have my hand. The hand has to pass on the signal to the brain. Suppose I put my hand in hot water. The hand passes the signal to the brain. Right? Yes. So which means it makes use of sensory neurons. So sensory neurons are also known as afferent nerve fibers. Okay. So the impulse has to pass, the signal has to pass so that it takes the information to the brain, right? So what it does? So we are talking about, so this is the structure of your neuron, right? So the signal has to pass through the entire length of the axon, that is the entire structure of neuron. Right? So this is conduction. So conduction is the transfer or movement of impulse along the length of a neuron or along the length of an axon. So impulse has to come here and pass on like this. So how it is going to pass? Okay. Hi Jigyasha, it is you. Yes, welcome to my session. That is so sweet of you joining here. So thank you so much. So please do like, share and subscribe Jigyasha. Okay. Fine. So how it is going to be done? So the first thing, understand the membrane of your neuron is known as axoplasm. So axoplasm is polarized. So polarized means it is charged. How come the axon membrane has charge? How does the axon membrane has charge? Because it has ion channels. So these ion channels are selectively permeable. Okay. So when the axoplasm is in the resting state, the axoplasm, yes, so the axoplasm has more K plus ions and more Na plus ions on the outside. So thus this leads to a lot of positive charge on the outside and a lot of negative charge on the inside. So there is a difference in charge, right? So this difference in charge is known as a concentration gradient. So this difference in charge is known as concentration gradient. Alright. So now how is it going to happen? So Jigyasha, are you understanding something? Yes, are you understanding something? Fine. So what is this concentration gradient? So concentration gradient is the difference in charge. So what enables this concentration gradient? Your sodium and potassium channels. For every 3 potassium goes out, so 2 potassium goes inside. Right? So this is sodium potassium channel. So this gives the difference in concentration of your axoplasm. So this is what is written in this slide. So the slide that we spoke about, for every 3 sodium that moves out, we have 2 potassium that goes inside the cell. So it is ideally like this. So this is our axon. So axon has more amount of positive charge on the outside, negative charge on the inside. So this is in a state of rest. Suppose if an impulse has to knock this position A, impulse has to pass through the position A, a signal occurs on this position so that the membrane permeability changes. So when I give an impulse here, the membrane permeability changes in this area. So it has more Na plus outside and K plus inside. But when I give an impulse, the membrane permeability changes. Na plus comes inside, K plus is about to go outside. Okay. So as a result, the charge also changes. Positive charge comes inside, negative charge goes outside. So this condition is known as depolarization condition. So in the resting state, it is in polarized condition. So when an impulse is applied, it is depolarized. Okay. Fine. Now, since the Na plus is coming in and the charges are reversed, the osmolarity in the axoplasm has to be maintained. Once again, the axoplasm should be stable, right? So quickly K plus moves out, okay, to restore the permeability of the membrane, right? But this permeability, the change in permeability is short-lived, okay? So now quickly the impulse passes through A and now it is ready to pass through the next section. If I give an impulse here, the membrane permeability changes. Na plus on the outside moves inside, the negative charge on the outside moves outside, the negative charge on the inside moves outside, and the positive charge comes inside, okay? But this membrane permeability is done only for a short time, okay? So this impulse is known as nerve impulse, fine? So now K plus goes out to restore the action potential of the axoplasm. Fine. So quickly the impulse is transmitted. So now what is resting membrane potential? So resting membrane potential is the potential of the membrane in the resting state. Potential of the axoplasm in the resting state. So this is your resting membrane potential. So this value is minus 70 millivolt. Fine? Okay. Now when an impulse is applied, so in the resting state I have resting membrane potential. So when I give an impulse, so that membrane potential on the inside and outside, that is known as my action potential. Okay, so along the neuron, the signal moves in this direction. Along the membrane, it moves in the opposite direction. So once this membrane permeability changes, it goes back to this state. So this is known as repolarization because the next region is activated. Because this is activated now. Okay, so this is the conduction of impulse. So I will continue with transmission of impulse in the next class. Okay. So next, when are we meeting? We are meeting on Wednesday. So Wednesday at 5 p.m. we have a mega mentee with 3 chapters. So please prepare well. The mentee is going to be a bit tough. So please share the link with all your friends and ask them to come. And please take the mentee seriously, because in a small population you should know where you stand. So that will give you the fear, the determination and the sincereness to go forward. Alright? Yes? So please prepare well for the mentee. I will meet you on Wednesday. So until then, have a great weekend. Study well, stay healthy and please be relaxed. There is no point getting frustrated. So this is me wishing you more and most if you genuinely like my way of teaching. So please do like, share and subscribe and bring your friends as well. Yes, thank you so much. So take care and stay tuned. Bye.